In the scientific method, researchers follow a systematic approach to investigate phenomena, formulate hypotheses, conduct experiments, and draw conclusions. Various types of variables are considered throughout this process. Here are the key types of variables to be considered. Independent variable. The variable that is deliberately manipulated or changed by the researcher in an experiment. It is the cause or input that is believed to have an effect on the dependent variable. Example: in a study investigating the impact of different fertilizers on plant growth, the type of fertilizer used is the independent variable. Dependent variable. The variable that is measured or observed in response to changes in the independent variable. It is the effect or outcome that is influenced by the independent variable. Example: in the plant growth experiment, the height or health of the plants would be the dependent variable. Controlled variables, constants. Factors that are intentionally kept constant or unchanged throughout the experiment. By controlling these variables, researchers ensure that changes in the dependent variable can be attributed to the manipulation of the independent variable. Example: in the plant growth experiment, factors such as sunlight, temperature, and soil type may be kept constant to isolate the impact of the fertilizer. Control group. A group in an experiment that is not exposed to the independent variable. The control group provides a baseline for comparison to assess the impact of the independent variable on the dependent variable. Example: in a drug trial, the control group receives a placebo, inactive substance, while the experimental group receives the actual drug. Experimental group. The group in an experiment that is exposed to the independent variable. The experimental group helps assess the effect of the independent variable by comparing its outcomes to those of the control group. Example: in a drug trial, the experimental group receives the active drug being tested. There are two types of controls: positive and negative controls. Positive and negative controls are essential components of experimental design. They serve as benchmarks for comparison and help ensure the reliability and validity of the experimental results. A positive control is a group or condition in an experiment that is intentionally subjected to a treatment with a known outcome expected to produce a positive result. It serves as a reference point for confirming that the experimental system is working as intended. Example: in a diagnostic test for a particular disease, a sample known to contain the disease may be used as a positive control to ensure that the test can detect the presence of the disease. A negative control is a group or condition in an experiment that is subjected to the same experimental conditions as the treatment groups but is expected to produce no response or a negative result. Example: in a drug trial, a group receiving a placebo, an inert substance with no therapeutic effect, may serve as a negative control. This group helps determine whether any observed effects in the treatment group are specific to the drug being tested. In summary, positive controls help verify that the experimental conditions are capable of producing the expected result, while negative controls help assess baseline levels and rule out the possibility of nonspecific effects. Including both types of controls is crucial for obtaining reliable and interpretable data in scientific experiments.